a greeting in Cherokee before.
Hello, everyone. So, welcome to our third live interview of the SSI Live Cosmic Conversations interview series. My name is Sophia, and I am one of the science communication interns here at Cal Academy. In this series, we're going to interview a different member of our SSI intern cohort every episode. SSI stands for the Summer Systematic Institute, a research experience for undergrads or an REO funded by the National Science Foundation. All of the interns that Nian and I will be interviewing work directly with curators or researchers on research projects on all sorts of topics, from plants to people to arachnids today. Feel free to leave any questions in the chat, and we'll try to get to them all at the end of the episode. Today, I'm going to be talking with Emilio, who's visiting us from Planet Arachnid. Emilio, would you like to introduce yourself before we begin?
Of course. Osio, dankadoa ia ustiona tsitalagi. Hello, everyone. My name is Emilio Chavez, and I'm Cherokee. And this summer, I'm working with Dr. Lauren Esposito in Arachnology. I'm looking at scorpion vomit over the summer, so that should be pretty fun. Looking forward to that.
Very cool. Thank you, Emilio. I'm very excited to learn more about scorpion vomit today. So why not, we'll just jump right into the questions. So first, can you tell us a little bit about, what is arachnology, and tell us a little bit more about the scorpions that you're studying this summer?
Of course, so arachnology is essentially, as the name states, kind of like the study of arachnids, and arachnids is this whole branch on the tree of life. So you have eukaryota, which are like mammals, and any eukaryotic organisms, and within eukaryota there's arachnids, there's spiders, scorpions, ticks, which I've had experience with very recently. I'm not too fond of that. In addition to crabs and other species as well. And I'm not exactly looking at a direct specific species, I would say, for my project. I'd say more of a region down in Baja California in Mexico. I'm working with a researcher, his name's Eric, and he's on a field station with the Academy of Sciences down there. And he's been collecting scorpions to collect their regurgitate. So he's basically making them puke, and he purified the DNA from the vomit, and he's trying to figure out more about their diet with some non-lethal sampling. Well, kind of non-lethal. We're still working on it. It has a good.
What do you mean by kind of non-lethal?
Right now, it has right now there's about 100 scorpions that have been collected. About 36 of them have actually, willingly given their regurgitate.
Okay.
And out of that 36, I'd say roughly about a third survived so far, which is, hang on, hear me out.
I didn't say anything.
Pretty much, unless we tested these methods, it'd have 100% mortality rate. In order to get their stomach context, we have to euthanize and dissect them and take it out of their stomach directly.
Mm-hmm.
So having a 33% survivability rate is quite a lot better than having a 100% mortality rate. So we're making a little bit of progress.
Very cool. What makes these specific scorpions, you mentioned they're from Baja California, what makes them special to be studying?
I believe that they are special to be studying. I know Eric, that's his key species of research right now. He hasn't sent me a lot of articles on it. I have to read about it myself, I will admit. Pretty much there are these medium, based on my knowledge so far, I'm still learning, they're like a medium-sized scorpion, and their diets and scorpions themselves have not been really documented that well and they haven't been researched to the extent of other arthropods like beetles and everything. So Eric, his goal, and my goal along with him, is to figure out the diets of these scorpions through the regurgitate. That way we could apply this method to other scorpion species as well, that we could better understand the entire species.
Interesting. So it sounds like, yeah, it's a pretty like understudied group. That's cool.
Yes.
So can you tell us a little bit more specifically about your summer research project as it relates to these arachnids?
Of course. So essentially what I'm doing is I'm given these purified samples of DNA that have come from Baja California. And over there, Eric, he's basically done what we call DNA extraction. You put in a variety of buffers and chemicals, and we take all of the DNA from the scorpion vomit, and we get rid of the byproducts. So we're left with essentially this big soup of DNA. And it's funny to say, but it's pretty much a soup. Everything's messy. There's no correlating, anything there. Nothing's correlated. And basically what I'm doing is I'm doing a method called PCR, just polymerase chain reaction. And I'm amplifying specific regions of the DNA that correlate to individual organisms. So it's called the CO1 region. And pretty much I'm amplifying those individual regions, which basically allow me to identify what's there and what's in the soup. And we're amplifying those regions using PCR. And then I'm testing them for their viability using a method called electrogel-phoresis, which is pretty much where I put the DNA into a gel and try to see if there's anything in there when it's electrified. I just did it today, got no results, so got to figure out what's wrong. So a little funky, but I'll figure it out. And from there, I'm going to send it to be fully sequenced through Illuminin sequencing. And I should get a full genome of the soup of DNA. And then I will use databases with already known genomes. And I'm going to do something called BLAST. And essentially that's when I put the whole genome into this program. And BLAST basically flags it for known genomes, sequences of known genomes from targeted organisms. So I'm going to have to discuss what these target organisms are for their genomes that will be used and how I'm actually going to use the program itself. Right now, I have to do the PCR and the electrophoresis for about 36 samples. Oh, wow. And I can only really do batches of 8 or 9 at a time at the moment. I just want to put all my attention in all of them to avoid, this happening and me wasting all my body.
Well, that sounds really interesting. It sounds like a very complicated process. So I'm curious to see how that evolves, as the summer keeps going.
Oh, of course. Believe me, I am too.
Very cool. So can you tell us a little bit about what might be like your earliest or one of your favorite memories you've had in nature?
Ooh, I'd say, so my favorite memory in nature, I'd have to say, is a long time ago, about 10 years ago, I'd say. So 10 years ago, I had my first involvement in science and nature at the same time, really. And it's also kind of where I got into arachnology too, kind of like a triple story here. So pretty much a long, long time ago, I went to this facility called the Hopland Research and Extension Center when I was little. I was 9 years old. Mm-hmm. And I was going there for a public program they had. It was called BioBlitz. And I met this wonderful lady there. Her name's Hannah Bird. And I met another man named Robert Kiefer. And funny enough, I also met Lauren Esposito.
Oh, wow. Mm-hmm. You guys go way back.
Way back. She doesn't know me, but I know her. Pretty much shout out to Bob and shout out to Hannah. Love you guys both. You guys helped me so much with everything in my life. Gotta say that. Pretty much when I was there, they were having a triple class, over the course of a couple hours. And it was on bats, astrology, and arachnology. And of course, Lauren was teaching arachnology. She was taking groups out into the woods with her black light to find scorpions by flipping rocks and watching them glow. And that was, I gotta say, I remember it so clearly in my head I remember walking up the trail and watching Lauren flip over rocks and finding scorpions and holding in her hands to everyone to see. And I thought it was just so cool. And I thought it was so amazing to see, actual science being done. And I've had experiences in science prior to this, but I think this really solidified in my head that I could actually do this as a job and I can make a living out of it. And not just arachnology, but any field in science. And I think that's really how it spoke to me in both nature and science, I'd say, my favorite memory. And it's also the first time Lauren got stung. That was
Oh no.
a little funny, not gonna lie. She described it as a wasp sting, so that was interesting.
Have you ever been stung by a scorpion?
Not yet.
Not yet. It's in the future.
Yeah. Further testing needed.
Yeah, I guess most of the scorpions you're working with are in DNA form, so they don't have the ability to sting you, I guess.
Got a lot of soup to work with. No stingers. Yeah.
Interesting. So you kind of already answered my next question, but I was, wanting to ask what inspired you to pursue science?
Ooh, well, I'd say definitely that one incident in BioBlitz really urged me to continue.
Mm-hmm.
I'd say there were like a few people who really got me into science. And it was my mom. She is a very, very smart woman. I was homeschooled for quite a long time, and she was my main teacher, essentially. And she was teaching me science a lot. She was teaching me a lot of biology. She's in the medical field. Mm-hmm. And she had this old microscope. I remember this microscope because it has a bunch of pieces missing and it has the lights broken, but you're still able to see through it. So every week or so, every day, we'd go out and we'd find something to look at under the microscope. And this is when I was in like 6th grade. And every time we found something new, we'd look at it under the microscope, like we'd be holding flashlights at different angles, trying to see anything. And it was always so cool and so fun to be able to be there with her and to experience those things with her. Because even though she knew all these things, there's some things we were both learning for the first time. And I just thought it was so heartwarming, and some of my best memories with my mom were formed through that. And I just really want to inspire others through that way too. And I try to do that through a lot of outreach, but that's really what got me into it.
Very cool. Thank you. So, how do you feel that you mentioned like your relationship with your mom and, visiting different places. Do you feel like your personal identity and like where you grew up influences your science or your research at all?
Absolutely. I grew up in a place called Ukiah. It's a small town. It's up in Northern California. And I was raised culturally. I'm both Native American, I'm Cherokee, and I'm Salvadorian. But as I was raised mostly to be in touch with my Cherokee side. And where I was growing up, there was also the Redwood Valley Band of Pomo Indians as well. And as I was growing up, as my mom's and myself and my whole family, they really want us to be involved in Native culture. And in Native culture, community is everything. Being involved with your community, talking to your community, helping. My brother and I were younger, we'd always go up and we'd gather willow for basket weaving. And my mom, she'd go out and collect roots that are used for, ceremonial practices. And we'd collect sage and we'd burn sage. And pretty much being in science I don't want to say it's taught me to be more in touch with my community. Being more in science, I see things that are sometimes done wrong with Native communities. A lot of the time, it's becoming more aware now, but it's just thinking of new ways to be involved with Native communities and to get Native communities involved in science. Mm-hmm. It's such a big goal. And whenever I go back home, I try to do outreach with Native communities as well with the Hopland Research Extension Center. I still volunteer there every time I go back. I take care of the museum. Cool. And whenever Native communities come up, I always go out to try to meet with them and to talk to them and to show them specimens and talk about science. And it's always been a main goal of mine to get more involvement there.
Very cool. It's really special, like, opportunity that you have to like connect those communities, and that like you're interested and willing to do that and like share, your interests with other people. That's really cool.
Absolutely, especially with museum specimens, you rarely get to see animals like that up close. And
Yeah.
you always see, like, you may see a coyote always running around your backyard, but you'll never get to pet one. But
Yeah, I feel like
close.
Yeah, like scientific specimens, I feel like are something that, like researchers and scientists are in touch with, but no, the general public is not at all. So that's really cool.
No. And there are cultural reasons too, because animals, they always have the spirits. And seeing one dead, it still has its spirit, and you want to take care of it. And that's what I really use for museum curatorial work. I always try to take care of the specimens the best I can, and always say prayers because I want to preserve the animal and keep its spirit alive.
Very cool. Thank you. So that is the last of our main questions, but I have a slideshow of photos of you. And so I'm gonna play them and see if you can like describe like the most important things in like 2 sentences for each of them.
2 sentences. Okay, here we go.
It's okay if it's not exactly, but
it was scary holding a scorpion for the first time. Thought it was gonna sting me.
Where were you?
I was over near Point Reyes. I can't remember the name of the specific place. We were there, and one of Lauren's members of the lab, his name's Luigi, went out and found a scorpion, and everyone was passing it around, and I just really wanted to hold it the whole time. And it was crawling all over me, and its stinger was like doing its thing. It was pretty cool.
You know what kind of scorpion this is?
That is actually, hang on.
Wave your hands around.
It was a forest scorpion.
There we go.
Oh, look at that.
Very cool.
That's actually a photo that is of me from the Hopland Research Extension Center. There's a small creek that goes right behind where I primarily am at. And it's called it's called, I can't remember the name of it right now. But it's a small creek that during the summertime and during spring has lots of flowing water. And after we finish doing field trips sometimes, the staff would go down there and put their feet in the water. And we'd all talk, and it's just really nice. And that is one of my favorite places to be. So as I said, to try to do outreach, and this is a photo of when I was back in Ukiah at the Mendocino County Science Fair. I was there with the Hopland Research Extension Center again, HREC. And I was out there with museum specimens from our collection, just trying to inspire and trying to do my best to just show what we have and to get the community up and close with specimens.
Very cool. Yeah, it looks like there's a really large range of different species on that table. That's really cool.
Oh yeah, each of them are prepared differently, and I try to teach the different methods we have for taking care of them. And that's a photo of my family. That's my grandma, my mom, and that's my partner, Montserrat. And they're all very, very important to me. Same my dad and my brother. They're not in this photo, but I'm always thinking of them too. But family is always very important to me. I do everything I can to be good with my family and to stay in touch with them. It's hard sometimes, but I always do my best for them. And I hope they think I do a good job.
They're definitely very proud of you.
Thank you. There's more outreach. There's the Hopland Research Extension Center. This was during COVID times, so I had to have that mask on me. Kind of nice to not have one on anymore. Kind of happy about that, But that was during a time when everyone was very much afraid to be out and about. And people who would come out to events, being there and just having a little distraction from what's going on in the world it's always nice and relaxing to me.
The first thing I noticed about this photo is the really large tree cutting behind you. Is that a sequoia? Or do you know?
That is actually, if I remember correctly, that is a redwood tree, because we're up in Northern California with these massive redwood trees. And I believe a long time ago that was cut down and transported to Ukiah, if I'm not mistaken. Might be wrong about that. I not too sure, but I'm pretty sure it's redwood.
Yeah, looks like it to me too.
And this is just a little example of some lab work that I do back in UC Berkeley. This is taken during another summer program that I had with my honors program called SEED, and it's for minority students getting involved in science. And if anyone's watching this tries, applies to Berkeley, I highly recommend this program. It's rigorous, but it gets you ready to be in science, and I cannot recommend it anymore. I have a lot of good memories from this program, and they still, provide a lot of benefits that I use today.
Very cool.
And this is a photo from when I'm actually working at Berkeley. I work in their museum. It's called the MVZ, the Museum of Vertebrate Zoology. I work there under a curator. His name's Chris Conroy. And if Chris, you're watching, I'm gonna send you a link to this. You gotta watch this. Pretty much, I am on a project called Ranges, and essentially it's a big project that's reanalyzing all the data in museums all across the country. And in the MVZ, we have to completely recatalog every single mammal specimen. And that's a lot of specimens, like 250,000. And wow. Back when we started, we had a team of 3. And it's a 10-year project. And over about the course of a year, we were able to get around 24,000 specimens done. And we were going at a pretty fast rate. And just a photo of me doing some fluid work with some ethanol mouse juice, That stuff that you gotta close easy, gotta tell you that.
My question was gonna be what species is in that jar, but you said mouse?
Deer mice. It's Paramiscus maniculatus. And there's all these deer mice there, just they're hanging out in this juice and they're floating around there. And I just take a look at their tags and make sure it matches with the database.
Very cool.
Lab coats for show, though. People in there, we wing it.
I think this is our last photo, last but not least.
That's a photo of a huntsman, actually. We were out doing on the same mountain that we got the scorpion. We found this huntsman, Luigi. Again, he found this huntsman. He's got some good eyes. Gotta ask him how he finds these things. But Luigi, he found this huntsman on the side of a rock, like on the underside.
Oh, wow.
And these guys, turns out they're not spiders, even though they may look like spiders. They're like a completely separate family. And they apparently only weave webs for very specific purposes, like to, relax and to hide. But they do active hunting through walking around and just finding things, which I thought was really interesting. And especially their legs, when you see a movie, their legs are kind of like hairs, like more like hairs than legs. They look a little disjointed and very, very thin. It's crazy to describe, but I just thought it was very cool. And I never saw one of these things before, or maybe I never paid close enough attention to see one.
Very cool. Well, so that wraps up. Thank you, Emilio. That wraps up our episode today. Thank you for joining us. And before we end, I'd like to encourage everyone watching to follow us on social media at calacademy_SSI. You can find us on Twitter and Instagram to learn more about science, natural history, and what all the interns are going to be up to this summer. You can tune in on Thursday. We're going to be interviewing Carol about some of her research this summer. Thank you, everyone, for watching.